Good morning, children. Now, in the last video, we had discussed about the Kelvin cycle. Here we are going to discuss about the C4 pathway or the Hatch and Slack pathway. Now, in some plants or in some crops like maize, sorghum, sugarcane, there is a different type of anatomy. Leaf anatomy can be seen, as well as they tolerate higher temperature. These crops, they have a special type of leaf anatomy and they also tolerate higher temperature. If such types of anatomy is there, we call it as Kranz anatomy. So, in C4 pathway, all the plants and the crops that are present, they show Kranz anatomy. Kranz anatomy, in which a special type of leaf anatomy is there, and those plants can tolerate high temperature. The first stable product that is formed is OAA. This OAA is a 4-carbon atom compound, and because it is a 4-carbon atom compound, it is known as C4 pathway. The leaf has two types of cells. The mesophyll cells and the bundle sheath cell. The mesophyll cell and the bundle sheath cells. So both of them combine to form the Kranz anatomy. During this process of C4 pathway, carbon dioxide is taken up and phosphoenol pyruvate is present in the mesophyll cells, which gets changed to oxaloacetic acid, that is OAA, in the presence of the enzyme PEP carboxylase. So, carbon dioxide is taken up and forms PEP, the presence of PEP, which is phosphophenol pyruvate. Where is it taken up? It is taken in the mesophyll cells. Taken in the mesophyll cells and gets converted to OAA, that is oxaloacetic acid. Gets converted to OAA. Now this oxaloacetic acid or oxaloacetate, this is reduced. This OAA, it's get reduced by maltated aspartate. Maltated aspartate, and then after getting reduced, it reaches the bundle cells. It reaches the bundle sheath cells. Okay. decarboxylation, this maltate or aspartate, it reoccurs by the release of during the process of decarboxylation, maltate or aspartate, it reoccurs by the release of carbon dioxide and formation of pyruvate. Pyruvate is a 3-carbon compound. So pyruvate is a 3-carbon compound. In high oxygen concentration, carbon dioxide, when the concentration of carbon dioxide is more, then rubisco, it behaves like carboxylase. When CO2 concentration is more, this rubisco it behaves like rubisco behaves like carboxylase and not oxygenase. It behaves like carboxylase and not oxygenase. In this process, there is loss of photosynthesis. So photosynthetic losses are, if it behaves like an oxygenase, then there will be loss of photosynthesis. But because rubisco does not behave like oxygenase, it behaves like carboxylase, so photosynthetic loss is prevented. Photosynthetic loss is prevented. Now RUBP. RUBP, it operates like Kelvin cycle. Operates like Kelvin cycle, and the pyruvate, that is the 3-carbon compound that had been formed, that is transported back to the mesophyll cells. Okay. So RUBP, it operates like Kelvin cycle, and the pyruvate compound that had been formed during this process, this is transported back to the mesophyll cells. Then they get changed or converted. When they come back to the mesophyll cells, here they get converted so that the PEP cycle, what is the PEP cycle? Phosphoenol pyruvate. So the PEP cycle continues. So these gets converted so that the phosphoenol pyruvate cycle continues. Now, why this hatch and slack pathway takes place in the C4 plants? So, how the C4 plants have been adapted? So, let's see about the adaptation of C4 plants. The first adaptation is that C4 plants, they lack photorespiration. Lack of photorespiration means photorespiration does not take place in the C4 plants. It shows high response to high light intensities. This means that they can survive even if the intensity of light is high. And they have great productivity of biomass. Great productivity of biomass. C4 plants, they also show Kranz anatomy. Now we had already discussed what is Kranz anatomy, which has the presence of the mesophyll cells as well as the bundle sheath cells. Why? Because two types of cells appear in it, the mesophyll cells and the bundle sheath cells. Now, they have presence of rubisco in the bundle sheath cells and PPKs in mesophyll cells. Presence of rubisco in bundle sheath cells and in mesophyll cells. In mesophyll cells. Their working is increased when the carbon dioxide concentration is near the rubisco cells in the bundle sheath. Their mechanism increases when carbon dioxide concentration is near the rubisco in the bundle sheath cells. So in this way, the C4 plants have been adapted to remain in such type of climatic conditions where even high intensity of light is present. So, and in this way they show the hatch and slack path. So I hope this is very clear to you. So this is all for today, children. So thank you so much.